I'm really excited to introduce Ryan Julian today. He's a professor of chemistry at the University of California, Riverside, where he leads a group centered around mass spectrometry since 2005. Dr. Julian obtained his PhD at Caltech under the guidance of Jack Beauchamp, focusing on a variety of molecular recognition and ion chemistry projects. He followed up with training in instrumentation and ion mobility with a joint postdoc with David Clemmer and Martin Gerard at Indiana University. After establishing his own lab, much of the emphasis has been on the development of structural tools, but over the last 5 years, a particular interest in the study of long-lived proteins and how they relate to lysosomal failure in age-related diseases has evolved. So it's, again, my pleasure to introduce Ryan today, and his title is Spontaneous Modifications in Long-Lived Proteins, Structural and Biological Implications. Take it away, Ryan.
Okay, thank you, Rich. It's a pleasure to be here and to be able to participate in this conference today. Even be better in person, but we'll do this remotely. Okay, so yeah, I'm going to tell you a story about long-lived proteins and some of our work in that area. I thought I would start out with this interesting slide here. Actually, I originally chose this for a different reason, but in the context of 2020, I realized this is one of the few things I haven't hit on the bingo list yet, so the 400-foot wave tsunami. All right, so let's see. There we go. All right, so lysosomal storage is a big part of everything that I'll be telling you about, so I want to jump right into that today. So basically what we're looking at is images of cross-sections within a brain, and the little particles that you can see in here are lysosomal storage. What's interesting, and that you probably are not very familiar with, is this is observed quite a bit in Alzheimer's disease. Actually, it's ubiquitous in Alzheimer's disease, just as it is in what they call the lysosomal storage diseases, which there are two examples of this right here. So this is actually a common feature of aging, and we're going to come back to this several times, so I thought I'd just jump right in with it. If we talk about Alzheimer's, you're probably more familiar with something that looks like this. So there are the intracellular plaques. Amyloid primarily consists of A-beta, which were originally discovered, and there are neurofibrillary tangles of tau. These are much more commonly associated with the disease. And in fact, the amyloid hypothesis is the main thing that's been pursued in terms of trying to treat Alzheimer's. And so the basic idea is that these deposits, mostly of A-beta, are the actual cause of the disease. This has been updated now to be the smaller soluble versions of this are the cause. But we can ask the question, you know, is that really what's the cause of Alzheimer's disease? And there's been really a lot of information along the way, both in support and also kind of to question whether or not it's the cause. And so this is some very recent work that I think is pretty interesting in relation to this. These are images, again, of the brain, and on the right, what's important here is you can see basically in the upper images the amount of amyloid plaque that was in this person's brain. And so here, this is actually somebody who has a genetic mutation that predisposes them to get Alzheimer's disease, and you can see that their brain is just absolutely hammered with plaque. It's very high levels. But in this case, they actually have another mutation that turns out to be protective. So this person actually did not develop dementia until 30 years after the normal date, and even when their brain had such high loads of amyloid. For comparison, this is the amount of amyloid that you would typically see in someone who already exhibited dementia with more typical kinds of Alzheimer's distribution of plaque. So this suggests that perhaps, you know, we should look into some other directions, and so that's one of the things that my group has been interested in doing. So the first slide I showed you suggested that Alzheimer's and lysosomal storage disorders are connected somehow. And this is actually not something that's new. This has been, those observations have been made for quite some time. So this is a nice chapter by Ralph Nixon, who's really done a lot of the nice work in this area. It was published over 10 years ago. So this is something that's known, but really, I point this out because there's still a question mark here, and it's kind of representative. There are still a lot of dangling questions as to how there is a connection between these things. So I'll just kind of give away a little bit of the answer. It may actually be related to chirality, but before we get into that, I'm going to take things back to the beginning. Actually, when I say the beginning, it's kind of like my beginning. And it relates to chirality and kind of give you an idea of how I got into this idea and way of thinking. So this is a mass spectrum, this is a mass spec conference after all, of the infamous serine octamer. And you'll either say, oh yeah, I've heard of that crazy thing, or what are you talking about? I don't know. So it turns out if you electrospray a concentrated solution of just the amino acid serine and look at it with a mass spectrometer, you get this very magic looking cluster of 8 serines. And so this was pretty interesting and drew a lot of interest from people. And so in graduate school, I actually studied this. And one of the things that we showed is that the serine octamer actually has a very strong preference for homochirality. So this is showing the amount of homochiral where we isotopically labeled the serines so we could tell which were D and L. This is how much should have been, how much was made in the homochiral version versus what you would have expected from a statistical distribution. So it really wants to be homochiral. And of course, it's known that life is largely homochiral. So some people actually push this so far as to say that serine octamer was responsible for the origin of homochirality in life. That was not a direction that I thought we could go with that. But I did get very interested in the kind of questions that were related to chirality and structure. And so to figure out why this serine octamer had this preference, you needed a structure that was both stable, low energy, and had kind of the required interactions of a chiral nature. This is the structure that we came up with, and this structure did really well. It was kind of the winner for quite a while. But it's been a time since I was in grad school. So 20 years and 65 papers later, people still been studying this thing. And actually just last year, a better structure came out with kind of quantum level calculations. And you can't really see what it is, but if you dig up your ACS calendar from last year, you'll find that there's actually a picture of the serine octamer in there. And so, so life is homochiral, or so we think, but if you actually look closely, there are a lot of examples where this is not the case. Now, if we're talking about proteins, you will actually find that there are D residues incorporated into certain peptides and proteins. And the initial and most common examples of this are actually in venoms and toxins found in these different organisms. So in this case, the D amino acids are actually intentionally incorporated. So there's a racemase enzyme that takes one of the initially L amino acids and flips it to the D form. And so you might ask, why is that? So there are at least 2 reasons that we know about. One of them is that in most instances, this inversion of chirality is required for the biological activity. So if you just look at the all L version of the peptide, it's not actually neurotoxic. But there's actually a second reason, which is likely why these have been incorporated, which is that it prevents proteases from degrading these toxins after they've been injected into whatever these things are trying to eat, basically. So we'll come back to that, kind of keep that in the back of our minds. So what about D amino acids in people? So there's no known instances in humans where D amino acids are intentionally created, but they are found. And they're due to spontaneous chemical reactions. So I want to really emphasize these are not enzymatically created, for the most part not even enzymatically controlled, and they can accumulate in long-lived proteins. So any protein that just hangs out for too long has a probability for doing this kind of spontaneous chemistry and flipping the chirality. Of course, this is the main context, or the main importance in the context of pharma. If you have proteins that are being stored, then these same kind of processes can, of course, happen. Within the human body, there's actually long-lived proteins in lots of different places, but importantly, they're in some kind of key organs, so the heart, eyes, and the brain have a decent number of these long-lived proteins in them. So one of the modifications is spontaneous chemical modification that's actually very difficult to detect, but is pretty important, as we'll see, is isomerization. So isomerization is one of these things, and basically it doesn't change the mass, it doesn't change the chemical nature of things, but it does change the structure. And isomerization is the most common and the most abundant example of isomerization. So basically we start here with L-aspartic acid, and the mechanism in this case is known. So you can attack the side chain and kick out water to produce this succinimide ring. This actually preferentially opens back up to the L-isoasp form, but it can also, the ring flip chirality, and then you can get the D-isoasp and the D-asp forms of this. So if you initially start with aspartic acid, you can end up with 4 different forms over time. You can do the same kind of chemistry by deamidation of asparagine, which you can track more easily because there's a mass shift associated from going from asparagine to aspartic acid. So this is just showing in an unstructured peptide the kind of distribution of the different isomers that you would get out of this. So L-isoaspartic acid will actually be the main thing that you would see, followed by aspartic acid, and then you do get some of the D isomers. Frequently, people ignore this entire side of the equation here, both in deamidation or isomerization, but as you can see, there are definitely some amount of these D isomers that are formed. So I just want to focus a little bit on the structure at the individual amino acid level. So serine, which I chose not because it was in the serine octamer, but because it's also the second most isomerizable residue in peptides. So if we start here and we're looking from the overhead view, looking down the backbone, you can see the side chain should be oriented this direction, but in D-serine, it's oriented on the wrong side of the backbone. In the isoaspartic acids, the situation is even worse. The backbone is elongated by 1 unit, it's kinked, and the side chains are both on the wrong side, one pointed up, one pointed down, relative to where they should be. So at an individual amino acid level, even though these changes are subtle in some ways and very difficult to detect, they do drastically alter the structure at a very close in view. Okay, so we're going to use mass spectrometry because, as I like to say, it's usually the best thing to answer any question that you might have. But in this case, it's a particularly tricky question, and it's even a tough one for mass spec because isomers don't change in mass. So we use a variety of instruments in my lab to do this, but one of the things that we have to do is modify them to do some photodissociation chemistry to give us a little bit of extra power for being able to identify these isomers. Fortunately now, you can also buy a commercial Orbitrap from Thermo that will do this UV-PD type experiment. All right, so what's the problem here? So as I already mentioned, isomerization doesn't change the mass. So if we look at these peptides, with one of them with an isomerized residue, they will have the same exact mass, not same nominal mass, exactly the same mass. So there's no way just looking at the full MS that you would really be able to tell anything about these from mass spectrometry. And so this is much more, a lot of you are familiar with PTM analysis, looking for phosphorylation and things of that nature. That's a much easier question. Even deamidation, you have a mass shift in all of those cases, so you can easily see it even at the full MS level. All right, so fortunately, there's other tools available to us, the mass spec, so we can do MSMS, we can basically fragment things and see what do the fragments look like. So this is a really simple example. These are actual results here of two isomers, and the fragments are over here, and you can see that the ratio of these fragments labeled A and B is changing quite a bit between the two mass spectra. This is typically what happens with isomers. You usually get the same fragments in all of the spectra, especially if it's just a stereoisomer. But if you have a structurally sensitive method, you can actually still tell that you will get difference in the abundance of those. So you can put a number with this. Typically what is used is called an R value, and it's just a ratio of ratios. It's the ratio of the two ratios of the fragments that change the most. Basically what you need to know is if that R value equals 1, your spectra are identical. If it's greater than 1, the higher the number is, it reflects that you had better discrimination between the two versions. So to be able to identify these isomers, one of the things that my lab did was develop something we call radical directed dissociation. And basically this is a fragmentation method that has a very high degree of structural selectivity inherent in it. And the reason for that is basically we can control exactly where we create a radical. So the first thing that we do is we append this group, and it basically is designed that when we hit it with a UV photon, it will very specifically cleave this carbon-iodine bond in a very high yield. And so that's what you can see in this mass spectrum. This is where the precursor was. It's mostly lost iodine. And then some of this is spontaneously fragmented into other pieces of the peptide. And importantly, these A ions are fragments that were directed by the radical. In fact, most of them will be. If we didn't see this fragmentation, say we had a larger peptide, then we can re-isolate this peak and add collisional energy to it and generate fragments that are still radical directed. So basically, because we've started this radical at this exact position on this arginine, and then it has to move to different locations in order to cause these different fragments, it's very structurally sensitive. And so it's a very good way of looking for differences due to isomerization. So I'll just give you one example here. So this is actually a part of A beta, and the serine is the residue which is known to flip from the L to the D form. So these are the spectra that we're looking at, and you can see that there's a very large difference between this -30 in the lower spectrum and the upper spectrum. And our chiral value for this is 31, which is quite a high number. I'll just point out one other thing. So these are very simple MSMS spectra. So a lot of times mass spectrometrists would be very bored by this because there's not very many peaks. And normally when we break stuff in a mass spectrometer, we want to see lots of peaks. That's what gets us excited. But for this particular application, that is not required. In fact, if you have a very simple difference like this, it's easier to see what's going on, and it's just fine. As long as there's a large change between the two spectra, it's fine if they're simple. So in this particular case for that peptide, if you look at the structures that come out of doing some simulations, you can see that the serine side chain actually tends to be oriented very close to that 4-iodobenzyl group that we've appended to the peptide. And so that's the origin. In this particular case, the -30 loss is associated with the side chain of serine, and so it turns out to be very sensitive in this case. In other cases, the R values may not be quite that large, but generally speaking, RDD is your best bet to be able to tell that there's two isomers there. So if we want to look at this kind of thing in a more complex kind of sample, say a biological tissue sample, then here's what we'll do. So first we'll get peptides or proteins out of that sample, and we're going to do just traditional LCMS kind of proteomics to identify what's in there. After we have a look at that, we will typically select some targets that we're specifically interested in, either because that's what we want to know about or we suspect they might be long-lived. And then we will make a hit list, so we will repeatedly look at any peptides that come up on our hit list over and over. And so this is typically what you might see for a peptide that's long-lived and had lots of different isomeric forms. In the chromatography, we'll separate it into some different peaks. Then we do MS-MS characterization to confirm that, in fact, those are different isomers according to the stuff I've just been going through. All right, and so you don't need any standards then to identify the isomers, the fact that an isomer is there. If you want to know the specific site of isomerization, then some additional kind of experiments will be required. So one of the first places that we actually started looking into this is not in the brain, but in the lens of the eye. And so the lens is a very interesting thing. Basically, proteins never turn over. So proteins that were in the lens of your eye before actually you were even born are still in your eye lens now, unless you had cataract surgery. And so there's a record of all of that, the history of those long-lived proteins. It's a good place to look. So if we examine the amount of isomerization as a function of age, as you can see here, just for one peptide, we can see that as we go to older people, there's an increasing amount of isomerization. And if we look at the same peptide within one protein, you can see there's some variation in this isomerization. So this box actually came from this data, and you can see there's this consistent trend where it goes up and up with age. In other cases, this peptide isomerizes much more quickly, so essentially by the time you're 40 years old, it's already largely isomerized. Another interesting thing that happens to the human lens is that it continues to grow throughout life. And so basically, like an onion, you keep adding more and more layers to the outside, and they are newer. And as you go down to the center, to the nucleus, you have the oldest material. So essentially, there's a history of the protein modification that's stored within the lens. So if we look at that within now a single lens, and basically, in this case, we look at the water-soluble cortex. That's the outer part. It should be the youngest material from the lens. You can see there's very little isomerization. The insoluble fraction will have older protein. You can see there's more isomerization. If we look at the nucleus, we can see even more isomerization. And if, again, we compare the trends looking at various peptides, all from the same crystalline protein in this case, there's a more consistent trend as it goes up in all of these, looking within a single lens. But when we kind of summarize things relative to the LCs that I was showing you before, it's clear that we've lost a lot of the details, and some of those details are actually going to be important for where we want to go next with this. So I'm going to pull them back out, and so now each one of these color bars represents the amount of one of the isomers that we found for each peptide in these different fractions from the lens. And so when we look at this data, it's complicated. There are lots of bars all over the place. You don't need to worry about it too much. I want to draw your attention, though, specifically to what's going on with this L-isoaspartic acid. So in the youngest fractions, we can see that it is not very abundant at all. And that's pretty interesting because, as you might recall from earlier on, L-isoaspartic acid in just a spontaneous chemistry system is the primary product that should be generated by isomerization, and yet its abundance is very small here. And if we keep looking at more examples of this through this protein and other proteins in the lens, we can consistently see that in the youngest fractions, L-isoaspartic acid is quite suppressed. So there is actually an explanation for this. So it's most likely due to the presence of this repair enzyme, PIMT. And so just to give you an idea of what that is, so here's the structure. Basically, it's a methyltransferase, and so it takes SAM and transfers this methyl onto the damaged site in order to try to repair it. And so it recognizes L-isoaspartic acid quite well, about 1,000 times worse. It can sometimes hit D-aspartic acid, and it doesn't do anything for D-isoaspartic acid. So just to give you a couple more of the details for this this enzyme, basically what it does is it methylates this site here, the unnatural side chain of L-aspartic acid. And when you methylate this, basically it drives this back to the succinimide ring very quickly because you have a good leaving group now. And at that point, PIMT, as far as we know, is done. So now we're back just to spontaneous chemistry, which if you're paying close attention means this ring will mostly open back up to go right where you started it. And then some of it will go back to the right L-aspartic acid, and a smaller fraction will also go down this pathway towards the D-isomers. So basically, PIMT pushes you this way, most of it comes back, it methylates again, it pushes you back, back and back. You may be thinking, wow, this has got to be the worst enzyme that's ever been created. Well, I'm not the best judge to say that, but whether it's an efficient enzyme or not, it's a very important enzyme, and that's been shown in studies with knockout mice. So if you eliminate PIMT, which has no other known function, basically the mice don't live past 60 days. So it's inefficient, but it's very clear from this that isomerization of aspartic acid, if left unchecked, is a very dangerous kind of situation. And is that timescale even make any kind of sense in terms of isomerization? The answer is yes, it does. So these are some studies that we did on the isomerization of A beta, the first part of it, which actually has the aspartic acids that isomerize most easily, and then a couple of the full-length sequences. Basically, what you can see is within 30 days, you have an appreciable amount of isomerization at both sites. So, around a little bit under 10% at the two sites. So, getting close to 16% at both sites. Within 60 days, you're going to be even higher. Not a ton of work has been published on this, but these values in pink come from various literature studies. So basically, isomerization of aspartic acid is not super fast, but it's definitely occurring within that kind of 50 to 60 day time scale that was very bad for the PIMT mice. Okay, so the bottom line then that we can take from this is long-lived proteins are dangerous when they're modified and not repaired. And so now the question we need to get into and come back to the beginning of the talk, does that relate to lysosomal storage, and if so, how does it relate? So if you haven't thought about this for a while, I'll just remind you of what we're talking about. So autophagy and lysosomes are closely connected to each other. So autophagy means to eat oneself. And basically in this process, old proteins, misfolded proteins, even entire old organelles that no longer function properly will be targeted, wrapped up into an autophagosome, and eventually fused with a lysosome where they're supposed to be degraded back into kind of building blocks. The lysosomes are quite busy because also anything that comes in through endocytosis eventually ends up fusing with a lysosome for the same kind of purpose. So basically, there's a very high flux of stuff that's being thrown at the lysosomes. They have a lot of things to do. It's not just proteins; all kinds of molecules are typically broken down. So in order to understand the connection with the lysosomal storage disorders, we need to understand a little bit about how those work. So again, this is the typical functioning state that we would talk about. So we're going to have a cargo of things that need to be broken down, which includes proteins, lipids, oligosaccharides. There's an entire suite of 50+ hydrolases that will break these down. So basically, if this is to happen appropriately, then you get small pieces that are recognized by transporter proteins and kicked back out of this fused lysosome, and you can kind of rinse and repeat. All right, so this is actually a very important part, this transporter. So it's easy to get into the lysosome. It's not easy to get back out of the lysosome. And that's kind of where the problem hits with the lysosomal storage disorders. So there are a variety of causes. They're all genetic, but the most common one is that one of the hydrolases is defective and doesn't recognize or break down its substrate. So basically what this means is it doesn't ever get broken down into pieces. The transporter can't recognize it and kick it out. And so at some point, the cell will recognize, okay, this thing doesn't work anymore, and it just stores it. Okay, the storage is from it just kicking it off to the side and make a new lysosome and try again. All right, so basically, this tells you how we get to the lysosomal storage and these lysosomal storage disorders. It doesn't tell us still quite how we get to lysosomal storage problems with Alzheimer's, but we'll get around to that. All right, so coming back to Alzheimer's connection. So we know that there are some proteins that are very strongly associated with the disease. So tau and A beta are both very involved. Certainly they're the main components of these fibrils and aggregates that are observed. So what is the frequency, or what do we know about the isomerization and epimerization that happens in these sites? So epimerization is if you flip from the L version of serine to the D version, that's a special subclass of isomerization. So for A beta, here are the most common sites that we would expect to see in red and blue for isomerization or epimerization. In fact, it's known that there is some extent of this observed in at all of these sites in actual A beta extracted from human brains. Tau is more of an open question. It hasn't been studied nearly as much, and the isomerization of it's almost essentially not been studied. But if we look at it, it looks like a minefield. There's lots of red and lots of blue in here. This is part of the reason why it can be hyperphosphorylated. There's so many serine residues. And then the proline threw in, it is also something which is difficult in terms of proteolysis, which is where we'll go in just a minute. Okay, so lysosomal proteases then are supposed to break down proteins that come in. And so how do they handle them? So just to remind you, if this were a protein, the mechanism of action, we need to get down to individual amino acids because that's what the transporter proteins recognize for the lysosome in this case. So cathepsin D is the most abundant lysosomal protease. It's one of the most important ones, and so that's where we started with some experiments just in a test tube. So basically, we've taken this peptide, the normal version of it, and we incubate it with cathepsin D, and basically this is what should happen. So we see lots of fragments of our peptide, and there's a very small amount of the original precursor left over. If we make just a single modification to this peptide and flip this normal aspartic acid to be L-isoaspartic acid, essentially cathepsin D is unable to digest this peptide at all. So even the small peak that you can see here is just oxidized methionine. There's no digestion. So just to report some of these results in a little bit more condensed fashion and show you the effect of other isomers, we'll show the data like this. So all of these colored short bars here represent peptide fragments from this sequence. And so this corresponds to the data that's up here. A long black bar will be a significant amount of the undigested precursor. So this data corresponds to this data up here. So you can see whether we have D-aspartic acid, D-isoaspartic acid, or even if we go to these serines, which are on the flanking sides of this peptide, there is significant interference and prevention of the proper digestion of this particular peptide by cathepsin D. So these are all cathepsin D. So cathepsin L is actually a much more aggressive cathepsin. It's responsible actually for 40% of all of the protein digestion in the lysosome. And so you can see for the normal peptide, it really hammers it. Lots of the fragments we don't even detect because they've been just obliterated. But again, once we start looking at the isomerized versions, the amount of digestion goes down significantly and is influenced in every single case. So regardless of which isomer we put or the location where we put it, it has a pretty significant influence on whether the two most important cathepsins can actually digest it. So there are actually at least 10, maybe 12 or more different proteases within the lysosome. And so there are both endopeptidases, which I just showed you, and exopeptidases. And so in an exopeptidase, basically the protease comes from either the N-terminus or the C-terminus and starts clipping off one or two amino acids at a time, kind of nibbling from the ends. So basically, the bottom line is we've also looked at cathepsin H and B, the two most common exopeptidases, and they can't penetrate within 3 or 4 residues inside the core of a peptide that has a site of isomerization in it. Actually, I want to show also some of the raw data that came from this just because it's kind of interesting. So this is the data that we looked at to check the exopeptidases. And one of the reasons I show that is all of these peaks in pink are actually endopeptidase activity. So we've done quite a number of protease studies in my lab lately. And so we've discovered that a lot of the specificity or site information for proteases that was figured out in the 90s is not quite right. The analytical chemistry's come a long ways. So just FYI, if you're studying a protease, you might want to check where it cleaves on your own. Okay, so similar results are found if we look into actual peptides or sequences that come from the brain. So A beta 1 through 9 again contains the aspartic acids at 1 and 7 that are subject to isomerization. And what's interesting in this case is if you look at the normal version, which is in black, so cathepsin L obliterates it. This is the amount of precursor that we're showing here, but cathepsin B still has some, and H and D basically don't touch it at all. So interestingly, the N-terminal part of A beta is kind of refractory to digestion just in general. But if you include isomerized sites, then basically it's totally dead. You can't digest it at all. So these are some sites in a sequence of tau where deamidation had been observed, so there would definitely be isomerization. And again, it's the same story. I don't want to belabor too much, but basically anytime there's an isomer, it's going to interfere with the digestion at that location. So there's actually a very simple explanation for this. The job of a protease is pretty challenging. Peptide bonds don't actually want to be broken down. They're designed to last a very long time, right? Remember, in the lens of your eye, those proteins last your entire lifetime. So this is what the active site with the peptide bound into it of cathepsin L looks like. And all proteases are going to have something very similar to this. So there's a channel. You need to bind the backbone in there with specific hydrogen bonds in a very particular orientation. You need to exclude water largely from the situation here. And then you can get your active elements to come in and initiate the cleavage of the peptide bond. So if we take, this is the recognition site here, if we take that and invert its chirality, in this case, you can see that now the side chain would be projected into the wall of the protease of the active site, all right? So basically, this is why digestion doesn't happen for an epimerized residue, because you cannot now have this here, so this comes out, and then you can't maintain the bond that's going to be cleaved in the active site. The situation is even worse again. So this is L to D. If you go from aspartic acid to isoaspartic acid, then we have this kink. The hydrogen bonding is all reoriented, and so if you try to fit that into there, you can just trust me, it's not going to work very well. All right. So basically, we know that isoepi modifications are going to prevent protease actions in the most common cathepsins, but then there's always, well, maybe there's another enzyme, maybe there's another cathepsin or whatever that's going to work if you throw this at biology. So that's what we actually did. So here we've made a peptide that's going to tell us whether or not it gets digested. So we put a cell penetrating sequence to get this into the cell and into the lysosome. And then we have a donor acceptor chromophore pair And basically, if the linker sequence where we can incorporate an isomer gets cleaved, then the fluorescence of this donor will turn on, and otherwise we won't see it. So we did these experiments in microglial cells, and so here we can see that the penetrating peptide works well. We can get it in there, and we can also verify that it gets into the lysosomal system. So this is where the peptide is, and then we use LysoTracker, and these are overlapping very nicely. So our peptide is getting in there and it's being delivered to the lysosomes. Now we can put multiple versions and see how does the relative amount of digestion in those different systems pan out. So basically, we have far more digestion for the native L version of the peptide linker than if there's a diiso. And so it does not appear that there's any magical protease in there that's going to efficiently digest isomerized residues in the lysosome. So you actually don't have to just take our word for it. There's been a variety of other people who have done experiments in this area and have provided supporting information. So this is interestingly work out of the Alexander Makarov lab, and no, it's not the Orbitrap Makarov, it's a different Makarov. And they were actually looking at isomerization of aspartic acid 7 in A beta. This is not related to any lysosomal experiment that they were interested in. They were doing this for another reason, but obviously it's still relevant to our interests. So basically what you'll notice is when they fed these cells isomerized aspartic acid, it was not good for cell growth. So A beta, normal A beta, was better than isomerized A beta. It was actually either the same or worse for cell death and worse for membrane damage. They also have done some experiments in different mice models. And so in this case, what they're doing is injecting isomerized A beta into the mice early on, and then they're going to wait some amount of time, and they're going to see whether or not this had an influence on the amount of amyloid plaques that were later observed in these animals. And so what you can see is after 4 months, the isomerized A beta had a much higher response, and after 10 months, this still persisted. So basically, in this experiment, it appears that isomerized A beta is significantly worse than the normal version. There could be multiple explanations that could account for this, but it's definitely interesting. This one is actually more difficult to explain in the absence of the amyloid kind of hypothesis. So in this case, they did the same experiment. They injected some isomerized and normal A beta, but they only injected 1 through 16. So 1 through 16 does not include any of the amyloid forming regions of A beta. And so it's more difficult to suggest that it was seeding the amyloid that was later observed in these animals. But it would be consistent with it basically causing difficulty with lysosomal digestion, and that would then be the initiating event that caused other problems. All right, so basically we have experiments in isolation, in cells and mice. All of these things suggest, including the PIMT knockouts, that isoaspartic acid is toxic. And so now I want to come back to where we initially started and kind of try to connect this back to Alzheimer's disease and other age-related disease as well. So in the lysosomal storage disorder, we had this substrate, and we typically had this defective hydrolase that doesn't work. And so this is what led us to the problem because we couldn't digest that. So we know that in Alzheimer's disease, this is not the explanation. So the genetics show that the hydrolases of the lysosome should be functioning in most people as it was intended to. But what I've just demonstrated is that there can be changes that happen to the substrate, so isomerization in the case of a protein, which then will prevent the normal hydrolase from being able to digest it completely. This will not be recognized by the transporter protein to get back out of the lysosome. And so perhaps on a different time scale, but with the ultimate same end result, you can get to a lysosomal storage situation where, again, this undigestible substrate accumulates to too great of an extent within the system. All right, so if you followed a lot of the Alzheimer's research, then you'd be familiar with some of the differences between A beta 1 through 40 and 1 through 42. And to a large extent, the experiments and information that relates to these two are some of the primary drivers of the amyloid hypothesis. Okay, so basically it's been demonstrated that if you are genetically prone to generate more A beta 1 through 42 than 1 through 40, you will get the disease earlier, have greater severity. At the same time, A beta 1 through 42 is known to be more aggregation prone. And so basically that's what's driven a large part has driven the amyloid hypothesis. So how does that fit into this whole lysosomal picture? Well, it's actually a very ironic and bizarre twist that it turns out A beta 1 through 42, but not 1 through 40, is a cathepsin D inhibitor, essentially. So it binds very tightly to cathepsin D and has a very slow rate of being digested. So essentially, you would expect that if you had more A beta 1 through 42, you're going to have greater sensitivity to lysosomal digestion problems involving isomerization, because you are already having some of the more crucial enzymes, namely cathepsin D, being inhibited. So this is work that Malcolm Lesring has published down at UC Irvine. All right, so if we start to try to put this together into some kind of picture of what's going on, then this is the way that we like to think about this. All right, so in a normal situation, we want cells to be operating under proteostasis. And so basically that means that the protein synthesis and the protein degradation need to be nicely balanced. And both of these are going to be proceeding at a very rapid clip, right? So then what we think happens is basically as a function of aging, we have this lysosomal capacity, which is a significant part of the degradation side of proteostasis. Initially, everything's fine. But at some point, if we start having accumulation of these isomerized to epimerized residues, perhaps other things contribute to this as well. You're decreasing the lysosomal capacity of the cell systems. And I should mention that this is lysosomal storage is almost always associated with post-mitotic cells. So this is the this is the place where this is going to manifest. You can't dilute out or have access to other mechanisms to deal with this problem. And the same is true in the lysosomal storage disorders. So at some point you have not enough protein degradation capacity, and so you now lost the balance between synthesis and degradation. This is actually a misrepresentation of what happens because as protein degradation slows down, it regulates protein synthesis to some extent, which will also slow down. So actually both of these begin to not function properly. And if you have neither synthesis nor degradation, then your cell is not going to last very long. So proteostasis basically gets disrupted. Okay, so that's the basic idea for what we think is the problem. At this point, people always want to know, okay, well, what's the solution? Please tell us there is some kind of solution. So we do have ideas along those lines as well. So let me just explain that. All right, so basically, if we generate new protein after some kind of short time scale. I've thrown in 2 weeks, but with an asterisk because that's just a totally made up number. You want to be kicking those new proteins into the autophagic system so that they get recycled before anything bad happens to them. And so we want to stay over on this part of the system where we have kind of robust autophagy that is recycling our proteins before they've lasted, say, a longer time scale that's too long. We have too much isomerization. We end up getting modifications. This now prevents the lysosome from being able to degrade them. We can't kind of get back over to this direction. And eventually, if we have sufficient accumulation of this, we end up in a lysosomal failure and storage situation. All right, so is there any evidence that that any of this is correct? There actually is, and so I'll take you through these results. So if we want to trigger autophagy more frequently, we have a couple of options that are known to do this. So fasting is actually a very strong inducer of autophagy. If you do pretty severe calorie restriction, this is also a method of triggering autophagy. In the context of this conference, there are definitely many companies that are looking into drugs that are able to trigger autophagy, stimulate autophagy. So I should also mention that fasting and calorie restriction in terms of aging, you may have heard, there have been numerous studies that show those have life-extending properties in terms of model organisms. That's not quite what we're looking at in this data here. So here we're looking at an Alzheimer's mouse model. Take whether you can really make an Alzheimer's model with a mouse or not. But basically, these mice overexpress too much A beta, and so later in life they exhibit difficulty. Neurological difficulties that are presumably similar. So what we're looking at is just a normal mouse, and then for the model mice, these ones were allowed to eat ad libitum, so they could eat as much as they want of the normal mouse chow. These were put onto calorie restriction, and these were put onto intermittent fasting. So these mice actually eat just as much as these mice, but they only eat it every other day. And if we look at basically then the behavior of these mice, they're all normal on a short time scale after 10 months, the blue bars. But as they age, basically the ones that eat whenever they want start declining, whereas those with calorie restriction and intermittent fasting are largely protected from whatever's causing this. So basically, both of these trigger autophagy. In theory, that should keep you over here, which would be consistent with the theory that I've presented you today. All right, so that's pretty much it. Just to sum up, long-lived proteins, I think, are pretty interesting. They've been ignored a lot. So the usual assumption is that if you lose function of a protein, then this is no big deal if you just make another protein and you're all good. But maybe long-lived proteins are still problematic because of their persistence if they change into a form that you can't get rid of. I've shown you that isomerization and epimerization are two of those kind of changes that may be likely to lead to those kind of problems. Remains to be seen, but increasing the frequency of autophagy or perhaps different people's level of basal autophagy may explain some of the differences between people that we see so far. So I'll finish up by acknowledging my group, the people who've done a lot of this work. So Jana Lyon did the initial work in the eye lens on the crystalline proteins. And then Dylan and Tyler are the two students who've mostly worked on the lysosomal data and the cell imaging. And we've gotten some funding from NIH and NSF to look into these things. thanks for the opportunity to come and present this talk. I'll be happy to answer any questions.
Thank you, Dr. Julian. Thank you very much for that great talk. Extremely interesting. I was wondering what your results suggest for potential avenues for treatment, or what, as you look forward, are there specific areas in lysosomal storage disease and in Alzheimer's in particular that are promising, and just general comments?
Yeah, so yeah, from like a pharma perspective, so once you have these, I mean, we don't know everything about these plaques and neurofibrillary tangles in terms of how long they're, do they have any turnover? How long are they sitting there? But we did do experiments that isomerization in the case of A-beta continues to occur even after you have fibril formation. So, so in terms of treatment, it may be a difficult question because the problem is probably already there. I think the better strategy may be prevention. So if this is an important part of the question, we need to like determine what's the level of autophagy that kind of prevents this from happening, and kind of make sure that that's being stimulated before you get to like dementia in the case of Alzheimer's.
Excellent. Thank you. I see we have some questions now from the audience, so I would like to transfer over to those. The first question is, how confident you are detecting isomerization by MS-MS ratio of the two peaks that are showed?
Confident? So yeah, we have looked at this quite a bit, and in highly controlled systems where we synthetically make the different isomers. So for example, in the crystalline system, we've made all of the isomers because it's not a vast system. So we know what the MS-MS spectra are for the de facto molecules, and they're, they've matched up. So we've done the statistical analysis to know what's the threshold of that R value that tells us the isomers are there. So that's one piece of information. I kind of didn't go through all the details. We actually also perform all of these experiments two ways. So we do just traditional collision-induced dissociation and look that way. Then we add our photo tag and we do RDD experiments, so we have that information. And then the second thing is, we also just, a lot of these separate by liquid chromatography. And so if they have the same nominal MS-MS spectra and they're not, eluting at the same time, there's not very many explanations you can come up with for what it could be. The usual thing people point out is, how do you know this is not cis-trans proline isomerization? And so we actually just recently published a paper on that, showing that in an electrospray mass spec experiment, it is exceedingly difficult to retain cis-trans proline isomers that came out of a separated chromatographic system.
Interesting. Excellent. Thank you. Another question from the audience. It seems like long-lived proteins are required in some parts of the body. Has any thought been put into how we might increase the autophagy pathway in a targeted way to avoid breakdown of needed long-lived proteins?
Yeah, so that's a great question. So there are lots of long-lived proteins. I don't know that they are recruited into the autophagic system as a natural course of things. So I think for the most part, similar to ubiquitin labeling for the proteasome, defective things, it's not just everything that goes, from what I understand, which you're getting a little bit outside of my area here, but that's definitely something that you would want to know about and think about. I don't know of any efforts to specifically trigger autophagy in like one cell type, or whether that be possible, be the short answer.
Okay. Thank you. An additional question, what MS methods do you recommend for detecting L and D asp and isoasp if RDD is impractical?
So, well, you can buy a new mass spec. That's always fun. If you really can't do it, it just depends. You may have to get a little bit lucky. It depends on the isomer. So we do all of these by CID, which is the most common, MS method. And sometimes you can still detect them from the MS. I would say you can also be more confident if you make the isomers and see the separation time. Retention times, obviously that's another avenue that doesn't require RDD. It depends on if you're scanning or if you're trying to answer one specific question, right? ECD or ETD also can give some unique fragments for isoaspartic acid in particular. So that is an option. It just depends for your particular peptide whether those differences happen. So the bottom line is, I guess I would suggest throwing everything you have at it and hope one of those things should stick.
So I think we have time for one more question. And so the question is are human genetic mutations of PIMT related to aging associated diseases? Are there substrate amino acids or domain selectivity for cathepsin B and L?
Can you repeat that?
Yeah, so I think maybe there's two questions here. So are there human genetic mutations of PIMT related to aging associated diseases?
Yeah, so, I have not encountered any studies of that kind of nature. PIMT is a little bit of a understudied enzyme, I think, but that doesn't mean there couldn't exist.
Okay.
I haven't seen them.
Okay, great. All right, so with that, I think that wraps up our keynote session today. Please join me in thanking Dr. Ryan Julian for delivering an outstanding talk. And I would also encourage everybody to please stay tuned. Our next topic up is our technical seminar that is sponsored by SciEx. The title of that seminar is Are Off-the-Shelf Formulation Buffers Suitable for Biotherapeutic Protein? The Effect of Buffer Formulations on Antibodies, Proteoforms, and Relative Abundance. So please stay online. That will be occurring shortly. Thank you.